Greetings to all. Myself Rhema Susan Varghese, Assistant Professor, Renal Dialysis Technology from Yenepoya School of Allied Health Science. So, today I am going to present the topic Principles of Hemodialysis. I have some specific learning objectives that are that at the end of the section you can be able to define dialysis, diffusion, osmosis, ultrafiltration and convection. Describe the role of the semi-permeable membrane in dialysis. Apply the principles of diffusion and osmosis to explain how waste products are removed from the blood during dialysis, and also identify the factors that affect the efficiency of dialysis. Before going to principles of hemodialysis, I would like to discuss about what is hemodialysis, when it is performed and why it is necessary. To understand this concept, we need to know what is kidney disease. We all know that kidney plays major role as excretory organ by performing functions like removing waste products from our blood, removing excess water from our body, production of hormones, keeping our electrolytes in balance, etc. Our kidney performs several functions in order to maintain balance in our bodies. If any reduction in their ability to perform these all functions, this will be leads to kidney disease or renal disease. What will happen if the kidney fails, or what will happen if kidney is not working properly or kidney got injured, what will happen? So the waste products will be accumulated in the body, and the normal composition of the blood will be disrupted, and even the patients will get complications, and it can be even leads to death. So there are two types of kidney disease that are acute and chronic. Acute means it occurs all of the sudden. It can be occurs within hours or days, and if we treated them correctly, it can be cured. It can be back to a healthy kidney. But in chronic kidney disease, it gradually occurs. It gradually affects the kidney, and it affects all kidney, and it is irreversible. So those who are, it will be lifelong. So those who are having CKD or chronic kidney disease, they have to undergo renal replacement therapy. So there are different renal replacement therapy, that are dialysis and transplantation. In dialysis, there are two types of dialysis. One is hemodialysis and peritoneal dialysis. So I am going to concentrate on what is hemodialysis. Hemo means blood, and dialysis means separation or filtration process. In hemodialysis, blood is removed from a patient body and circulated through an extracorporeal circuit that is outside the body, and it is cleaned and returned back to the patient's body. In this extracorporeal circuit, there is an hemodialysis. You can see in this figure, there is a dialyzer in the circuit. So this dialyzer is also known as artificial kidney. So it when the blood which are taken out from the body, it is cleaned in the dialyzer and returned back to the body at the same time. So metabolic waste or toxins are filtered from the body by the semi-permeable membrane and carried away by the dialysis fluid. The goals of hemodialysis are to manage uremia, fluid overload and electrolyte imbalances that occurs as a result of chronic kidney disease. This is the working of a hemodialysis. You can see that from one artery, the blood is taken out and it is circulated in a extracorporeal circuit. From the dialyzer, it is cleaned by the dialysate, and clean dialyzer is entered to the dialyzer, and the waste dialysate is removed and it is returned back to the body. So in a dialyzer, there are two compartments, blood compartment and dialysate compartment. So through through one compartment blood will flow, the other compartment the dialysate will flow. In middle there is a thin layer called semi-permeable membrane. Through the semi-permeable membrane, this principles occurs, that transport of molecules or solutes solvents, etc. are done. So we are going to discuss how it is happen. So principles of hemodialysis. To know about principles of hemodialysis, we have to know about like solute, solvent and solution. Solution means it's a combination of solvent and solute. Solvent means it will be a fluid form, and a solvent is a substance capable of dissolving any substance, like a substance called solvent, and a substance like solute, and a solute is substance that dissolve in a solvent. For example, in a salt water, salt will be the solute, solvent will be the water, and salt solution will be the solution. In dialysis, dialysate is the solution, treated water will be the solvent, and solutes are the electrolyte present in the dialysate solution, that like potassium, sodium, chloride, etc. and glucose, etc. So you can see that there are slight changes in the concentration of blood and dialysate. For example, in blood the concentration of potassium is 4.5 milliequivalents per liter, but in dialysate it's 2 milliequivalents per liter. We match electrolyte levels in a dialysate closely to the patient normal blood level. This is why we are doing like this. This means to reduce the loss of electrolyte out of the blood during dialysis. So the next is semi-permeable membrane. As I said before, in a dialyzer there are two compartments, blood compartment and dialysate compartment. In the middle, you can see in the figure, in the middle there is a thin layer of material is there, and with a pores or opening that allow a certain substance means like molecules, middle molecules or small molecules to pass through freely while keeping other substance out. Through this, blood cells are not pass through it, because the blood cells have a large molecular size. So through this, waste products, excess chemicals and excess fluid can pass through it. So there are four principles, that is diffusion, osmosis, ultrafiltration and convention. So we are going to discuss each one in detail. Diffusion is the process by which solutes move across a semi-permeable membrane from an area where they are in high concentration to an area where they are in low concentration until the concentration are equal. When two solutions come in contact through a semi-permeable membrane, each solution have different concentration. So in the figure you can see that in one side it is represented as red, which is having high concentration, and the other one is represented as white and is having less concentration. But when the diffusion, when the solutes move across the semi-permeable membrane back and forth, the concentration become equal, like the solute movement will go on until the concentration on both sides of the membrane are the same. For example, we will take tea bag as an example. Tea bag acts like membrane. When you place a tea bag in a hot water, some of the water will be enter into the tea bag, and the concentration of the tea, like flavor of the tea, or will be high there, but outside the tea bag it will be less. Consider inside of the tea bag. So but when you steep the tea bag for a few minutes, the molecules from the tea bags will be distributed among the water until the tea is done. Then the concentration will be equalized, like will be equal after some times. So at this time the water inside and outside the tea bag contains the same concentration of the tea. The aim of the diffusion is to achieve equilibrium and equal distribution. By the process of diffusion, the waste products from our blood is removed through the dialysis. So, in the body, diffusion into and out of the cell goes on all the day along to maintain our electrolyte or maintain our body in balance. In dialysis, waste diffuses across the dialyzer membrane into the dialysate. This is how we remove them from the patient's blood. There are certain factors affecting diffusion. That one of the factor is concentration gradient. So in concentration gradient, solutes move from higher concentration to the lower concentration across a semi-permeable membrane. The difference is gradient. In dialysis, we create a concentration gradient between dialysate and patient's blood to force the solute to move out of the blood. We also use gradients to balance electrolytes levels in the patient's blood. For example, if the concentration of potassium high in blood, but the potassium level in the dialysate will be less, so the concentration will be, diffusion happens and the concentration will be equal, and the concentration of the potassium from the blood will be reduced. So the next one is molecular size. Smaller molecules diffuse more easily than the larger one. Then the next factor is temperature. Warm molecules move faster than cold one. In the figure, you can see that in one glass it is hot water and the one other glass it is cold water. The diffusion happen much faster in hot water than the cold water. So we in dialysis we keep dialysate at a safe temperature to maintain patient's comfort and allow faster diffusion. The next one is membrane permeability. A membrane with more pores allow faster diffusion. In membrane permeability, it consider like a number of pores, pore size, the design and the thickness of the pores. This all will be matter for the membrane permeability. A membrane thickness and design also affect the diffusion rate. The next one is surface area. Larger surface area allow more diffusion. And the next one is flow geometry. In flow geometry there are two types of flow geometry in dialysis. One is co-current and counter current. During dialysis, blood flow in one way and the dialysate flows the other side. That means that is counter current mechanism. In counter current mechanism, the diffusion occurs faster than the other co-current mechanism. In co-current mechanism means the dialysate and blood will be flows to the same direction. This mechanism mostly is we used for the children's. The next one is osmosis. Osmosis is the movement of solvent through a semi-permeable membrane from an area of lower solute concentration towards an area of higher solute concentration. So osmosis is another one of the nature ways of maintaining equilibrium. Same as diffusion, in osmosis also they maintain equilibrium, equal distribution among. So during dialysis we use osmosis to remove water from the patient body. How we are using osmosis? Osmosis for removing water. So we create a osmotic pressure gradient in the dialysate. That by change how we are creating osmotic pressure means we will change the solute concentration on two sides of the membrane. For example, we think about a jar. If we dissolve sugar in one side of the jar and the other side will be plain water, after some days we can see that where the sugar level, sugar water solution is there, the level will be increased compared to the plain water, because sugar is a absorbent of sugar or glucose will easily absorb water. So if we dissolve sugar in a water on one side of the membrane, the osmotic pressure draws water across the membrane surface, diluting the sugar water. Eventually, the water level on the sugar side will increase above that on the plain water slide. Adding more sugar increases osmotic pressure and rises the sugar water level. This is how we are removing excess water from the patient's body. So in diffusion, solutes moves. In osmosis, the solvent moves across the membrane. In both diffusion and osmosis, movement goes until the concentration become equal on the both side of the membrane. This is the osmosis and diffusion happening in the dialyzer. Ultrafiltration, the movement of solvent across a semi-permeable membrane caused by a pressure difference is called ultrafiltration. By the process of ultrafiltration, the excess fluid from the patient's body is removed. How we are removing excess fluid from the patient's body is that we will give an pressure to the blood compartment by the help of dialysis machine, so that the pressure will squeeze water into the dialysate, while blood compartment pushes water across the membrane into the dialysate compartment, draining the solute. So we are applying a pressure which helps to squeeze the water from the blood to the dialysate compartment, and the dialysate will be removed, and also the excess water also will be removed. The factors affecting ultrafiltration. There are certain factors affecting ultrafiltration, that is ultrafiltration coefficient, that means the water permeability of the membrane. And the next one is transmembrane pressure. Transmembrane pressure is nothing but it's the pressure given in the dialyzer between dialysate compartment and the blood compartment. And the next factor is membrane pore size or the surface area, and the next one is rate of flow. The blood flow and the dialysate flow also affect the ultrafiltration rate. If we increase the blood flow rate and dialysate flow rate, the ultrafiltration will occur more. Then the next one is negative pressure on the effluent side. Several parameters determine how much fluid needs to be removed during dialysis. Includes the patient estimated dry weight and actual pre-treatment weight, and also as well as how much fluid will be added during the treatment process. For example, if the patient is undergoing saline dialysis, how much saline is giving to the patients, that amount should be removed from the patient's body. So we will add more ultrafiltration rate to the more ultrafiltration rate. How we are setting ultrafiltration rate means it's a dialysis machine only engineered according to the to very precisely to control the amount of fluid that is removed from the patient bloodstream. The dialysis therapist only need to enter how much amount of water should be removed from the patient's body. Just enter into the dialysis machine. The machine automatically will calculate how much should be removed from each hour and how much pressure should be given, and all the settings will be automatically calculated by the dialysis machine. And the last one is convection. Convection is the transfer of heat and solutes by the movement of the parts of a gas or liquid. In dialysis, convective transport leads to a solvent drag. During the ultrafiltration or by the pressure given for the ultrafiltration, the solvent also dragged through the water. solute also dragged to the water. As a solvent crosses a semi-permeable membrane, it drags along smaller solutes. Those solutes that can pass easily through the membrane pores are swept along with the water. The ease with which the solute is dragged along is determined by the size of the solute molecules related to the size of the membrane pores. So, this is the systematic representation which is happening in the dialyzer. From the dialysate and blood compartment, which all should be removed, like urea, creatine and like nitrogenous waste are removed from the blood, and the bicarbonate are taken from the dialysate to the blood. This is happening in the dialyzer. And the summary of the summary, there are 4 principles of hemodialysis, that is diffusion, osmosis, ultrafiltration and convention. What is diffusion? Means movement of dissolved particles or like solutes across a semi-permeable membrane from an area of higher solute concentration to an area of lower solute concentration. The next one is osmosis, movement of fluid across a semi-permeable membrane from an area of lower solute concentration to an area of higher solute concentration. The movement of solvent across a semi-permeable membrane caused by a pressure difference is called ultrafiltration. And convention is the transfer of heat and solute by the movement of parts of a liquid or gas. Convective transport in dialysis leads to solvent drag. And these are the references in the Vancouver style, and thank you.